Hello everyone, my name is Laiba Khan and I'm currently an undergraduate student at Georgia State University. Today I will be giving a short introduction to my poster presentation, How FGF Morphogens Can Lead to Monogamy. The concept of survival of the fittest describes the process of natural selection and eventually reproductive success. It is scientifically established and understood that numerous environmental conditions, adaptations, and outside influences can affect how a creature's offspring survive. Parental care, which is common in monogamous relationships, is one specific aspect that can lead to an offspring's increased chance of survival. Although many animals form socially monogamous pair bonds, a biparental convict cichlid provides a good non-avian paradigm for understanding monogamy. The lateral hypothalamus has typically been known to be labeled as the feeding center and the pleasure center due to the different types of neurons that it contains and the impact that they have on maintaining homeostasis. It is developed by various proteins, and one of the types is a family of morphogens called fibroblast growth factors, FGF. FGF plays multiple roles in neurogenesis, exon growth, and differentiation, and these morphogens play an important part in the development of the lateral hypothalamus and are critical for normal development to occur. An example of a morphogen that is key to the development of the ultimate structure of the midbrain and hindbrain, where the lateral hypothalamus is located, is FGF8. The 9 amino acid neuropeptides, such as oxytocin, are good candidates as modulators of pair bonding because they exert widespread influences on affiliative behaviors across a variety of vertebrate classes, in addition to modulating many aspects of physiology, including vasoconstrictions, hydromineral balance, and smooth muscle contractions. Oxytocin is primarily produced in the hypothalamus, where it is either released into the blood via the pituitary gland or to other areas of the brain and spinal cord, where they bind to their respective receptors to alter brain and physiology. Sexual intimacy can increase oxytocin production, which helps adults bond. The hormone isotocin, which is IT, which is a fish homologue of oxytocin, has been found to briefly modify mating behavior in convict cichlids. These effects have led oxytocin to be grouped with the other happy hormones known to have a positive impact on mood and emotions. In the central nervous systems, genes encoding oxytocin and its carrier protein are expressed in the hypothalamic supraoptic and paraventricular nuclei. The abnormal hypothalamic oxytocin system in the fibroblast growth factor 8 deficient mice study examined if fibroblast growth factor 8 is essential for the proper formation of the oxytocin system, and then these data suggest that a reduction in FGF8 signaling leads to an overall reduction of mature oxytocin levels that may have resulted from defects in multiple stages of the hormone synthesis pathway. Since proper hormone synthesis is a hallmark of mature oxytocin neurons, this study suggests that FGF8 signaling may contribute to the phenotypic maturation of a neuroendocrine system that originates within the diencephalon. Considering that most fish share DNA and brain structure, it's odd that convict cichlids have such distinct social behaviors, and to determine why this is the case, a series of tests were performed to identify and compare the expression of the FGF gene in convict cichlids' developmental stages to that of the zebrafish, Danio rerio. Throughout this procedure, my goal was to test my theory that the monogamous behavior observed in convict cichlid fish is due to increased levels of FGF signaling morphogens forming the hypothalamus, which promotes the production of oxytocin hormones. I will be discussing the difference between the gene expression in both convict cichlids and zebrafish in detail on Wednesday, 13th April from 10 a.m. to 11 a.m. If you want to know more about my research, or if you have any questions, feel free to drop by. Thank you so much.